All right, so this is the last tutorial lecture today. So we are very happy to have Professor Joachim Krug give a talk, and the title is Structure and Navigability of Fitness Landscapes from Models to Data.
Okay, yeah, thank you very much for the introduction and for getting us to this spectacular place. Also, I'd like to thank you for the way you have set up the day, because I think you'll see that the two previous talks, many themes will recur, and I'll try to maybe bring them together in a little bit from the perspective of physicists. I mean, physicists always have this fate of being halfway between mathematics and real science, right? I mean, all right. So this will be about fitness landscapes. And I should mention, this is work that has been funded in Cologne for a long time through various collaborative research centers. Currently, we have one on predictability and evolution. So let me give you an outline what I want to try to do here. I probably have more material than I can cover, but I think we can maybe return to some of these topics also later in the week. So I'll begin with a few slides on empirical fitness landscapes, right? So what are fitness landscapes, and what do we sort of see, what have we sort of learned about them from the empirical side in recent years? Then I want to give a very basic introduction to the mathematical framework that we're using. This will be done on the blackboard. And then I have sort of two, if you will, case studies on two different models or classes of models, which actually also have direct analogs in spin glasses, namely the random energy model, which we like to call the house of cards model, and Fisher's geometric model, which has an equivalence to certain types of Hopfield models. So for sort of further reading, so what I will talk about here and much more can be found in several recent review articles with Daniel Oros, with Sakshi Paujani, and currently there's a paper under review in genetics with Malvika Srivastava, Claudia Bank, and Suman Das, which sort of tries to give a fairly broad and up-to-date review of the state of the field. Okay, so since we're in Chicago, I obviously couldn't resist actually starting with this seminal paper by Sewell Wright, who at the time when he invented the concept of fitness landscapes was, of course, at the University of Chicago, right? So we're sort of,
when was that?
We have a kind of genius loci here. Sorry?
When was that?
1932, almost 100 years ago. And so he was the first one who was sort of imaginative enough at that time to imagine what the entire field of gene combinations would look like, graded with respect to adaptive value, right? So adaptive value is what we now call fitness. So the idea is that you have this space of gene combinations and you assign fitness values to them, and this is sort of the space on which evolution happens. And famously in this paper, there are two illustrations, and they already sort of tell part of the story. The first one is this combinatorial space of gene combinations, right? In 1932, of course, people didn't really know what DNA is, but they knew that there were entities called genes, discrete entities that were present in discrete forms, so-called alleles, and they were arranged in some sort of one-dimensional way. And so Wright here imagines what the space of 1, 2, 3, 4, 5 genes that can occur in two different combinations looks like. And this is, of course, the hypercube, right? And this is the hypercube that was already mentioned by Gerard, and that you will see much more of in my talk. So this Wright was very much aware of. But then there is a second picture. And so what he imagined was that on this space, you would have a random landscape, essentially, right? So that's going to be the main topic of my talk. But according to a biography that I read, the editors of this paper thought this was a too abstract a picture, so they said you should put something more intuitive, right? So he put this picture as well, where he now imagines the genotype space as being a plane, and he's now able to plot the fitness landscape as a kind of hill and valley structure. But he writes explicitly in this paper that this is a very inadequate representation, right, as we know. But he was fully aware of this. Over the next 100 years, or let's say over the next 70 years, so throughout the 20th century, I think this is the picture that stuck. Right? This picture very rarely appeared in the literature.
And unfortunately, the crucial phrase in there,
under a particular set of conditions
Obviously.
also seems to have disappeared.
I agree, but I will also ignore it in my talk, right? But yeah, I mean, he knew what he was talking about. Right. So this picture pretty much disappeared, but of course there were lots of debates about what these fitness landscapes were really like over much of the 20th century, when there was no way of testing these things experimentally. So famously, already in the 1930s, there was a debate between Wright and Fisher whether fitness landscapes actually had multiple peaks or not, right? So whether they were really, as we say today, rugged. This was, if you read it, this was essentially a debate about functions on continuous spaces versus discrete spaces. So even if there are multiple fitness peaks, it doesn't necessarily mean that there are lots of valleys and that sort of the evolutionary trajectories have problems crossing valleys. So that's to some extent a separate question. A general question, of course, that also came up today is, is it even meaningful to think about these hyperastronomically spaces which evolution can never explore? We'll always get only local glimpses of these landscapes. And so the question that I want to address today is, given this situation and given the difficulty of imagining these high-dimensional fitness landscapes, what is the role of models, right? And so what could be useful null models that can be used to interpret empirical data and to see whether, what kind of structures you can find in them. So that's sort of the point of view that I want to follow today. Okay, so let me give you a very short tour of empirical fitness landscapes. Arguably, I would say this field started about 20 years ago with this by now iconic paper by Daniel Weinreich and collaborators with this nice title, Darwinian Evolution Can Follow Only Very Few Mutational Paths to Fitter Proteins. So what these people did was to study all combinations of 5 mutations in a particular antibiotic resistance enzyme, which together increase antibiotic resistance by a factor of about 45,000, antibiotic resistance being quantified in terms of the concentration at which the bacteria can still grow. And so this picture is showing a subset of that combinatorial space. So here you have the wild type, so the original enzyme. Here you have the 5-fold mutant, and here you have a subset of the intermediates, which are denoted by this binary code with minuses and pluses showing the absence or presence of a certain mutation. And you see these arrows. And so what Weinreich was interested in was how many ways could this wild type evolve into the 5-fold mutant. If you just add one mutation after another, there are 5 mutations, so there are 120 directed mutational pathways, and out of those, only 18 are increasing in resistance, right? So if you follow a stepwise trajectory where in each step you increase resistance, this is only possible in a relatively small subset of these paths, right? So this certainly got me interested, and I think also a lot of other people in this field. And I think the important thing about this was sort of the image, right? The visualization. This is another fairly early picture from our own work. So this was the beginning of my collaboration with Arjan de Visser at the University of Wageningen. And this is, again, a 5-dimensional landscape. So this is now, the 5-dimensional hypercube in its full glory. In this case, the underlying biology are individually deleterious mutations in Aspergillus niger, a fungus, and they all reside on different chromosomes. And so that made it possible to combine them into the genome. And this was actually done by Arjan much earlier. So this data set is from 1997, so preceding Weinreich by almost 10 years. But this picture didn't appear in their publication because the only thing that they were interested in at that time was how fitness decreases as you increase the number of deleterious mutations. So they basically, in the picture, you will only find a kind of radial plot of this fitness landscape where you see how fitness declines with the increasing number of deleterious mutations. But this combinatorial structure and the complexity of it was not there, right? So that appeared only once we knew that we should look at this.
What is the color? What is green, red?
Right. So this hypercube now has an orientation. I'll get back to that. So the idea is, and this is a concept that was introduced by Christina, So now you have a fitness landscape on the cube, right? So now you can orient the graph in the direction of increasing fitness, right? So these arrows point in the direction of increasing fitness. And then you have these, a couple of these sequences where all the arrows are coming in, right? And they are the peaks, right? So you have one peak at all zeros, and you have a couple of more peaks here. So these are the maxima in the landscape, and the colors indicate that these, so the blue ones, for example, are in the gradient basin of this particular peak, right? So now you sort of see a kind of structure of evolutionary processes appearing here. So this was the beginning, right? So we studied combinatorial landscapes of relatively modest sizes. This is now 20 years ago, and of course the technology has improved tremendously. So nowadays the scales are rather different. This is a picture from a few years ago. This is a landscape that was obtained in the lab of Michael Desai. So this is now quantifying the affinity of the SARS-CoV-2 spike protein to the receptor in the human cell. So a kind of fitness component of viral evolution, if you will. And it's showing all combinations of 15 mutations separating the ancestral Wuhan strain from Omicron BA.1. So this is about 32,000. And the way this is plotted now is on this axis you have the number of mutations, so the distance from the wild type, and these dots here are the different fitness values. And you see that there is sort of on average, there's a kind of valley. So in the beginning, the affinity actually doesn't increase. An even more recent study from the group of Andreas Wagner is showing sort of what I would consider the state of the art in these kinds of combinatorial large-scale fitness landscapes. So this is sort of illustrating how this landscape is constructed. So it's a particular protein, DHFR, which also appeared already this morning in the talk. So here the authors decided to look at 9 nucleotide positions, so 3 codons in the FOL-A gene that were sort of expected to be relevant to antibiotic resistance to a certain antibiotic tree methaprim. And then they basically created all these variants, in this case about 260,000 of them using CRISPR-Cas technology. Then they ran a mass selection experiment and did sequencing, and that way they could basically estimate the fitness of all these variants, right? So in the end, you end up with a fitness values for these 260,000 variants, and then you can start, studying evolutionary questions. And I'd like to point out here that again, in this graphical abstract, what appears is right second picture, not the first, right? So again, they are thinking about this in terms of hills and valleys. So now you can ask questions about peaks. You can ask questions about reachability of peaks. And this is what they did. So for example, they find that this landscape has 514 fitness peaks, 73 of which have high fitness in the sense that the fitness is higher than wild type. You also have lots of peaks that have lower fitness than wild type. And so at this point, when I first saw this paper, at this point you could ask, okay, you have these 260,000 plus genotypes, you have 500 peaks. Is this a lot or not, right? I mean, this is sort of coming to the complexity question. What is sort of your null expectation?
How many of these are doubly stable to pair mutations? If you say it has to be stable to 2-point mutations, is that eliminating most of them, or do you still fail?
I don't know. But the more important thing is, and so you can do a simple calculation that I will also get back to. So this is sort of one example. I think you can see what these kind of null models are good for. So let's suppose that we assign fitness at random. So the first thing to notice in this paper is that there are 260,000 plus genotypes, but only about 18,000 of them are functional, right? So you have a relatively much smaller functional network, right? Now so these are the ones that we really should care about, because lots of these mutants, of course, don't really work. So let's suppose that these 18,000 functional sequences are in a sort of compact region of sequence space and form a complete genotype graph. Then we can estimate how many peaks there should be, because the number of peaks is then simply the number of sequences divided the degree of the genotype graph, that is the number of mutational neighbors of each genotype plus 1. I mean, this is a formula that was first derived by Kaufman and Levin, and I will get back to it in a moment, right? So this is a simple estimate, and then you get 644, which is not that different from 514, right? So if you believe this calculation, which of course has many, caveats to it, you would say, oh yes, this is indeed a highly rugged landscape. And this is sort of a strategy that, I'm proposing to experimentalists in general. If you have a landscape like this, you want to see if it's highly rugged, randomize your fitness values, just shuffle them and see if the number of peaks changes or not. Right?
Here's a basic question for connecting to Rama. So there are just 3 amino acids. So why aren't there just 8,000 relevant? I mean, for the steps, you need to worry about nucleotides, but for the fitness, you presumably don't.
No, you do. I mean, the synonymous mutations are not neutral. In general, they are not. I mean,
in the protein, they're just measuring the protein.
Yeah, but there's expression, there are other things. I mean, the
protein.
Yeah, but you're actually measuring fitness here, right?
In a cell.
Yeah. You're actually measuring growth rate of the cell. So no, but this is sort of a common finding that synonymous mutations are not. They typically have weaker effects than the non-synonymous ones, but they're not neutral. Now another. Yeah.
Another question, just to understand the numbers here. You were in dimension. I mean, you had 4 to the 9.
Yes.
And then some of these are not functional. 4 to the 9 is 2 to the 18. So, and the number of variable is 18 here. Your cube.
Sorry.
Your cube is in dimension n. A cube has 2 to the n vertices.
Yes, but the connections are different. It's not just a cube, right?
So that's what I'm asking. Is it nearest neighbor or?
Yeah, it's nearest neighbors, but it's nearest neighbor in the Hamming graph, right? So each sequence has, whatever this number is, 3 times 9, 28 neighbors, right? So it's not a cube, right? It's not a cube. I'll get to that. It has a different geometry.
But it's not really high dimension, right? It's between 9 and 29. It's not there yet.
It's 20. Is 28 a high number? I don't know. For a physicist, it probably is almost.
It's George Gamow said 1, 2, 3 infinity.
Exactly. Yeah. So in another study from the Wagner lab, they which was about a bacterial transcription factor, they also looked at so-called adaptive basins. So I'm mentioning this because this has been motivating for us also to study these adaptive basins. So what is an adaptive basin? An adaptive basin of a peak is the set of all genotypes from which the peak can be reached along a fitness increasing path, right? So this is just like in Gerard's talk. Imagine you have, some evolution going downhill, and how many genotypes are there that can reach the peak via a fitness increasing path. And the surprising finding in this study and also in other similar studies is that these adaptive basins are typically very large, especially if they are of high fitness. So in this case, the fitness is repression because this is a transcription factor. And you see that for the highest peaks, 30% or even 50% of all genotypes belong to one basin, right? So this space is enormously highly connected. And this is sort of a general observation that Wagner and collaborators have made. You can also see here a nice linear correlation of the basin size with a peak fitness rank. So the higher the peak, the larger the basin. And so this is a feature that we found recently you can actually explain, or you get the same feature in a completely random model, right? So this is something that I will get back to.
So these basins heavily overlap.
Oh, of course. Yeah. They heavily overlap. I should say, there are sort of 2 types of basins, right? So in the previous, oh, now I'm going the wrong direction. In the previous picture, so in this picture, I showed what are gradient basins, right? So a gradient basin is a basin that comprises all genotypes from which the peak is reached along a gradient walk, which means, or a greedy walk, right? Where you sort of, you do the, or steepest descent, right? You do the highest, you do the largest fitness increase each step. Those are, of course, disjoint, right? So each genotype belongs exactly to one gradient basin, but it belongs to many adaptive basins. So there's an important distinction. Okay, so I'll get back. This will be like the third part, but for now, I want to summarize a bit the basics for you. And I'll do that on the blackboard. So the first concept that we need to think about here, and that we have seen in the examples, is a concept of a sequence space, right? Right, so I'm looking at sequences, and I'm using, for some historical reasons, always the letter sigma for a sequence. So a sequence has a length, which is L. Right, so this is my sequence, and the elements in the sequence will be from an alphabet, and this alphabet is of size A. Right? So these are the things that are in population genetics called alleles. So each of these entries can take A different values, and there is a natural distance measure, which is the Hamming distance, right? So the distance between two sequences is simply the number of elements in which the two sequences differ, right? So this we can write in this way. All right, so this is the distance. And so the sequence is an element of the product of this set of alleles, right? And so this set of sequences together with this Hamming distance defines a graph, which is called a Hamming graph, right? So this defines a Hamming graph. Which we like to denote by H, all right? And for A equal to 2, these are the hypercubes, right? So for A equal to 2, we have hypercubes, which we have already seen in the lecture this morning, right? But in general, they are not. So for the nucleotide alphabet, you would have A equal to 4, right? Right, so let me just, draw a couple of pictures. So now I'm making a mistake here. This should be a capital A. So let me just, draw a couple of obvious pictures, right? So if we do this for the binary case, this would be our cube, right? And for 3, so this would be L equal to 2. This would be L equal to 3. So this cube is actually already an example that is good enough to illustrate a lot of things. So this is sort of what I will typically draw. What I want to mention here is that the geometry for the binary case and the general case is somewhat different. So importantly, for A equal to 2, you have each point has a unique what we call antipode, right? So you can for a given sequence, you can just define a sequence sigma bar, which has elements that are simply 1 minus sigma I, right? So like here, the all 0 sequence is the antipode of the all 1 sequence and so on. So, which means, and this already came up this morning. So if you now think about a very large hypercube, it has these two poles, or you can select these two poles, and then it has an equator where most of the sequences are, and this equator is at distance L over 2, right? So most sequences are at distance L equal to 2. So this is a sort of a useful way to think about about the space. Whereas, of course, when A is greater than 2, right, if you now want to have an antipode, so an antipode is a sequence that is at maximal distance from a given sequence, right? And if you have A possible elements, and then, of course, there are A minus 1 other elements that you could go to, right? So you have A minus 1 to the L antipodes for each sequence. Right, so it's not you shouldn't think of it as a sphere anymore, right? I mean, it's something that has sort of many poles, and most sequences are not at distance L over 2, but at distance A minus 1 over A. Right, so there isn't an equator in the middle, but the equator is a bit shifted away. Okay. So this is the space, right? This is the space that Wright drew and that we will be thinking about here. Now, the next important concept is this concept of fitness graphs that I already introduced. And so this term was introduced by Christina Krona and collaborators. So what is a fitness graph? So the idea is now, so a fitness landscape is essentially a function on this Hamming graph, right? Now, so I will call it G. So this is a function that takes me from this Hamming graph to the real numbers, right? And now I can do what I already indicated. I can say, okay, this function defines an orientation, right? I can simply draw arrows in the direction of increasing where this function is increasing, right? And that defines an orientation, which by construction is acyclic, right? So it's like essentially like the gradient field of a function, right? So this is an acyclic orientation. And so let me, draw an example here. So we have to put some arrows here. Right, so this would be an example of such a fitness graph. And now you can see that this fitness graph has certain structural elements that are of interest. So for example, we can ask where are the peaks, right? Because these arrows are always going in the direction of increasing fitness. So the peaks are the sinks of the graph, right? So the peaks are the points where all the arrows are pointing inwards, right? So we have one peak here and we have one peak here. So this thing has two peaks. And the other thing that we are interested in and that we will be studying in the following are these fitness increasing paths, right? So now if you want to study trajectories on this graph, then you need to follow the arrows, right? So for example, you can have a path like this, right? So this would be one possible path to this. And these paths, of course, always end in peaks, right? So this would be this yellow path, for example, ends in this peak, and you can now start thinking about how many paths are there, and how likely is it that you find paths of a certain length, and so on, right? So, and this is a concept that, that in principle one could, of course, have introduced in the spin glass literature, in the 1980s, thinking about energy monotonic trajectories. But in fact, this question, as far as I know, was first posed by Dan Weinreich and collaborators in the connection of this empirical study that I showed you. So this yellow thing we call an accessible path, right? So accessibility means that the arrows, that you basically follow the arrows, right? So these are accessible paths. And what you can see here is that there are sort of different types of accessible paths. So of course, if you think about a path between 2 points, the shortest path will cover exactly the distance between the 2 points, right? But the path can also be longer. So this is, for example, a path that in the end covers a distance of 2, but it has length 4, right? So there are indirect, there are direct and indirect paths. And this will become important later as well. Okay, so this is sort of the setup. Is this clear so far?
I have a question.
Yeah.
So in this representation, is there any way to like tell which peak is like higher peak?
No, in this representation, not. So this is, I mean, so a lot of what I will tell you, or basically everything that I will tell you, is only about order relations, right? So I'm only talking about these nearest neighbor order relations which define the graph, right? So that's additional information that you would need to add. Okay.
Sorry, for the gradient basin, you need to know, right?
For the gradient basins, you would need to know, right? For the adaptive basins, not, right? So the adaptive basins you can define on the level of this graph. That's right. Okay, so let me try to continue here, maybe. So the next concept that I think I cannot avoid discussing a little bit, and that we also heard about already this morning, is the concept of epistasis, right? So epistasis has something to do with a pairwise interaction between mutations, right? So let's look at the simplest example of a set of 2 sites, right? Sequence of length 2, binary sequence of length 2. And I have some fitness values associated with this. And so the question of epistasis is now about the relationship between the single mutations and the double mutations, right? So I can introduce selection coefficients. So for example, the selection coefficient of the first mutation in the background of the 0 string would be, in my notation, this, right? And the selection coefficient of the second mutation would be this difference, right? And so now the question of epistasis is, can I predict? So I know the fitnesses of these 2 types. Can I predict the fitness of the double mutant, right? If I can, then there is no epistasis, right? So what is the prediction? I mean, at this point, you have to, of course, define, what is your null expectation? Typically here, one assumes that the null expansion is linear, which then means that so the additive expectation is that, so let me call this G add of the double mutant, is that I take the single mutant, or I take the wild type, and I simply add the 2 selection coefficients, right? So that would be the additive expectation. And this you can then write as G of 1, 0 plus G of 0, 1. And then, but then you have to subtract G of 0, 0 twice. So you have 1 G of 0, 0. Right? And then, so if this is true, then there is no epistasis, and so the epistatic interaction is then the deviation, right? So this is basically the actual fitness of the double mutant minus the expected one, right? And this we can now write down. So this is equal to G of 1, 0 plus G of 0, 1 minus G of 0, 0 plus G of 1, 1.
Can I ask a question about this?
And just a moment. And you can see that this is actually a kind of curvature, right? So epistasis is essentially a curvature of the fitness function. Yes.
In experiments, you often don't have direct access to the fitness, and this is not invariant under nonlinear transformation if I use log of things. So could you comment on that? It's always kind of never understood what the right resolution to that is.
Neither have I. I mean, I think, you know, you can do this on linear scales. You can do it on log scales, right? If you have, of course, you could also use a multiplicative null model.
But like in measurements, just particularly when you want to apply it to data, like you measure like growth rate in Rama's experiments, and you know, maybe how is it correlated to the binding or something, right?
I mean, my general tendency is that, you know, growth rates, if you grow like in discrete generations, your null model should be multiplicative, right? So then you would apply this to the log of the growth rate. I think this is what most people do. But of course, for other phenotypes, you might not even know. I mean, there, you know, sometimes people say one should use different scales, but I think what I want to emphasize here is that, of course, you know, defining some particular pair of mutations as non-epistatic is probably going to be rather rare. I mean,
it's interesting mainly when two are the high and then the two diagonal ones are low. So
that's the point. I think that, you know,
then what a nonlinear transformation doesn't change.
Exactly. I think and that's sort of going to be the next point, right? So the more informative thing is actually the signed interactions, right? So this is okay, but this is just like the classical definition. Now, sign epistasis is making exactly this point about now just looking at the order, and this is again a concept that was introduced by Dan Weinreich. Right, so here you basically don't care about the actual values, but essentially you care about the fitness graphs, right? So sign epistasis is a property of the fitness graph. And so, you know, if there's no, and the way you can sort of see in the fitness graph if there's sign epistasis or not is just by looking at arrows at opposite edges, right? So for example, you know, this arrow here is shows the effect of a mutant at the first site in the background of 0, and this is showing the effect of the mutant in the other background, and if they have the same sign, then there is no sign epistasis, right? So this would be an example when there's no sign epistasis. There could, of course, still be epistasis, and this is then what Weinreich calls magnitude epistasis, right? And then there are the other two possibilities that you could have one pair of arrows being directed oppositely and the other still in the same direction, right? This is then called simple sign epistasis. Or you could have both pairs showing in opposite directions, but in such a way that there doesn't appear a cycle, right? Which means that I cannot just, you know, I cannot put the arrows here, so I have to put them like this, right? And so this is what is called reciprocal sign epistasis, because both of these are sign epistatic with respect to each other. And this little fitness graph now shows you that there now have to be necessarily two fitness peaks, right? Because in this graph you have two sinks. In the other graphs you only have one sink, right? And so that tells you that reciprocal sign epistasis generates fitness peaks. And there is actually a theorem. So there's a famous statement by Polvik collaborators showing that any fitness landscapes that has more than one peak needs to have at least one reciprocal sign epistasis motif. And this was generalized recently by two different groups to show that if you want to have n peaks, you need to have at least n plus 1, n minus 1 motifs of reciprocal sign epistasis. And one of these proofs used a discrete Morse theory. So this just as a comment to what was discussed this morning, right? So this is sort of a landscape with n peaks as at least n-1 reciprocal sign epistasis motifs. So that tells you that this is a kind of local condition that tell you something about the existence of peaks, multiple peaks in the landscape. Okay, there is a generalization of this epistasis analysis to more than two loci, things that go under the name of Fourier-Walsh expansion and so on, but I don't really have time to go into that here. If in my view, there has been a somewhat controversial discussion around this in the last couple of years. So this might be something, a possible topic also for a discussion later in the week, but I can't really go into it here. Okay, any questions so far? Okay, so let me now get to the first of the two models that I wanted to talk about, right? And this is the so-called house of cards landscape. So let me just briefly define what it is. It doesn't require much notation, really. So this is what we call the house of cards model. Which in this form was introduced by Kaufman and Levin. So you notice that 1987 was a bit later than Derrida's introduction of the random energy model. It's essentially the same, but you will also see when you read Kaufman and Levin, you will see that they discuss a lot the connection to spin glasses and so on. This was very much inspired by the spin glass development, and so very much like in the spirit of the lecture this morning by Gérard. The idea is, okay, we want to learn something about these, these functions on these sequence spaces, and we define some random model. Now, the simplest random model is simply to say the fitness values are IID random variables, right? So G of sigma is an IID random variable. It should have a continuous distribution so that you don't have any degeneracies, but otherwise the distribution actually doesn't matter, right? So because we're only interested in rank order properties, the distribution doesn't matter. Any monotonic distribution will not change the results. And the most famous and simple result that I already made use of in the previous part is to ask what is the probability in this setup for a genotype to be a peak, right? So let's compute that. So let me call this P max. So this is the probability that the genotype sigma is a peak. Right, so this is a probability that the fitness of sigma is equal to the maximum of what? Well, of the fitness itself and of its neighbors, right? Right, so each genotype has neighbors, and we're looking at this Hamming graph. So there will be, as I had discussed before, we have n equal to a minus 1 times L neighbors, right? And so the probability, so here we're basically looking at Here we're looking at n+1 random variables, right? They all come from the same distribution. So the probability that any one of them happens to be the largest is simply 1 over the total number, which is 1 over n+1, right? So this is the formula that I had used before. So this is basically a minus 1 times L plus 1. This is the probability to be a peak. And so the expected number of peaks then is the total number of genotypes divided by this, right? And so this is the number that in this comparison with the Wagner experiment gave us these 640 or so peaks, right? So this is sort of, and this, of course, this is just showing that, you know, many properties of this model can be simply derived from simple order statistic relations, right? And so what I want to explain to you next is this recent paper with Daniel Oros, where we computed the sizes of adaptive basins in this model, right? So now you have a peak, you know how many peaks there are. The question is, what is the size of a basin, of an adaptive basin in that landscape? And it turns out that this can be computed very explicitly. And I think the argument is essentially rigorous. This is just a recap of what I just told you. This is the House of Cards model. And so the results are based on work by a previous student, Benjamin Schmiegelt, who discovered a phenomenon that we call accessibility percolation. So what the question that we're asking is the following. We take this random landscape, we pick 2 genotypes which have some fitness difference, right? And we ask how many accessible, that is fitness increasing path are there between these 2 genotypes, right? So there can of course only be an increasing path from the lower to the higher genotype. So let's, you know, take 2 of these genotypes. I have a label L here, so this should actually be it. Yeah, no, I think that's right. I need a label L because I want to scale L to infinity, of course. And so let's denote the number of accessible paths from one to the other by Z sigma tau. And then the phenomenon of accessibility percolation is that when L becomes large, the probability of finding such a path has a sharp transition from 0 when the fitness difference is smaller than a certain threshold to 1 when the fitness difference is larger than a certain threshold, right? So there's a threshold beta C, which here, because all the fitnesses are in the unit interval, is also a number between 0 and 1. And this beta C is the root of a certain somewhat complicated function. I mean, the strategy is simply to ask, what is the average number of paths, right? So you can calculate the average number of paths, and you find that the average number of paths goes from below 1 to above 1 at this threshold. But then there's a lot of other work to prove that this is actually a sharp bound. But this is the case, right? And this depends on the number of symbols in the alphabet, and it depends on delta, which is the scale distance between the 2 points. Obviously, you know, if the 2 points are further away from each other, then this will also be less likely, right? So there's a dependence on this delta here, which is the Hamming distance divided by L. And now, and the insight that we had more recently was that this is, of course, in some sense already solving the problem of the adaptive basins, right? Because if I have a peak, I can now say which genotypes have a path to that peak, namely those genotypes whose fitnesses satisfy this condition, or rather, you know, so the genotypes that have a fitness below omega sigma minus beta C will belong to the basin, and those that have a fitness that is higher do not, right? So the probability that any genotypes belongs to this basin is simply this, right, in this uniform, on this uniform scale. Now, if these are peaks, then the fitness of the peak is also going to be very large, or, you know, large meaning close to 1, right? So a typical peak genotype has a fitness close to 1. So the probability that it belongs to its basin is 1 minus beta C. And then I invoke the geometry of the sequence spaces that I discussed in the beginning, which is that if I pick any peak genotype, most of the other genotypes will be at a scale distance a minus 1 over a, right? Just like so in the binary cases would be 1/2, but for in general it would be a minus 1 over a. So the delta at which I should evaluate this function is simply this delta star, right? So this gives us this result that the expected basin size in units of the total number of sequences is simply 1 minus beta c evaluated at a and delta star. And so this is a number between 0 and 1. And as you increase the number of alleles, this goes to 1 because the leading order expansion for large L is log a over a. And you can even calculate it explicitly for small a. So for 2, it's about 0.72. So this means that a typical basin has 28% of all genotypes. And for 4, for the nucleotide alphabet, it's 0.47. So a typical basin contains more than half of all genotypes, right? And this we also checked numerically. So this is showing simulations of these basin sizes for different a. So this is a equal to 2, 3, 4, 10, and 20, and different L. And you see that the results converge. We can show that the leading order corrections to this result are log L over L. So they're pretty large, but you can see that it sort of converges here from below. And this result, of course, also immediately gives you this linear correlation, right? What it also tells you is that if you now look at not necessarily peaks, but you just look at any or you look at atypical peaks with small fitness, then the basin, the probability for genotype to belong to the basin is simply linear in the fitness, right? So you immediately get this linear relation between basin size and peak size that I showed you in the experimental results of Wagner. Right, so this gives you a rather simple picture based on this very simple model. Of course, I'm not, and this sort of goes back to discussions that we had previously today as well. Of course, I'm not saying that these experimentalists are just measuring noise, right? That they might as well just replace all their measurements with random numbers. But what we are saying is that these large basins and the linear correlation between basin size and fitness is not something that you should be surprised by, because it can be achieved even in this very minimal model. Okay. Yeah.
So this is true of sigma that are independent, right? So if we state, the experiments say that if I add some correlation between different sigmas, so the fitness, this goes true. So do you have some perturbative?
No, we don't really. I mean, this is an obvious question, right? So what can we say? Obviously, the real fitness landscapes are not random. So what can we say if we introduce correlations? And that's maybe a good, let me maybe go to the next slide. So because what I wanted to do in the end is now also to talk a little bit about what we call structured fitness landscape, right? So suppose the fitness landscapes are not random, but they have some structure. So there are a couple of different models that have been studied in the literature, which sort of introduce these correlations in different ways, right? So one of them is the NK model that was introduced by Kaufman and Weinberger. So there the idea is essentially that each the contribution of a genotype to fitness depends on the states of a set of other genotypes, but not all of them, right? So there's a kind of interaction range here, which is called K. This is, I think, somewhat similar to the p-spin models in spin glass theory. Another one is the Raffman-Fuji model, where you take the random model and you add a kind of, what did you call it? A signal, right? So you add a signal essentially. So you put something, a deterministic variation with a distance to some reference sequence. So for both of these models, there are results on the accessible path. but they are fundamentally different. So my take at the moment is that if you introduce correlations, what happens to these results for the random model will depend very much on what correlations you introduce, right? So certainly these two models are not really similar. The third class of models that I want to, in the last minutes, want to discuss in a little bit more detail are landscapes which we like to call landscapes with global epistasis. So global epistasis is a concept that I think was also mentioned this morning. It's unfortunately a somewhat ambiguous concept because different people mean slightly different things with it. But what I mean by global epistasis here is that you have a landscape that is generated by a nonlinear phenotype fitness map. So you introduce a kind of intermediate level, right? So you say you have a linear function of the genotype, right? So in this sense, just the genotype, the elements with random coefficients, and then you apply a nonlinear function to that, right? So that's one version of global epistasis. In protein sciences would mean, for example, that you have some additive phenotype, but then, you know, if you study, for example, thermodynamic stability, then this will be mapped to fitness with some nonlinear function. So this is the mathematical structure. It turns out that this is actually closely related to what is known as Fish's geometric model. So this is sort of a slightly more detailed statement of the same thing. So we, in general, of course, this phenotype can also be multidimensional. So you can map it not just to one phenotype, but to a vector of phenotypes, and you can shift it, right? So you have some more elements. Important thing is that these landscapes can be, you know, the genotype landscape is typically multi-peaked. If there is one phenotype and phi is non-monotonic, or even if the phi is monotonic and there's more than one phenotype, then can also be multi-peaked. So what we're sort of interested in is how multi-peakedness and complexity is generated in these kinds of landscapes. Let me illustrate this maybe in a simple example, right? So let's suppose we have a one-dimensional phenotype, right? So this is my phenotype. We have 3 sites and we have binary sequences. So the linear phenotype is simply this linear combination of these A's, and these are the vectors A, right? So these are 3 different mutations that all have different effects, right? So now, I say fitness is now a function of this phenotype, which has a peak, right? So the idea being, you know, this could be the expression level of some protein, and some expression level is maybe optimal. So if you have more expression, it's not good. If you have less expression, it's not good. So there is a peak somewhere in between, right? So let's say this is my fitness function. And so now what I have to do is I have to generate, by this additive rule, I have to generate the phenotypes of the different genotypes, and then I have to map them onto fitness, right? And so when I do this, this is sort of what happens, right? So these are now the phenotypes of the different. So this is, for example, the phenotype of this particular sequence, and it maps to this fitness and so on, right? And you can now see this sort of cube being embedded here. And importantly, what you see is that in this way you can get multiple fitness peaks, right? So here you have again 2 peaks, 2 genotypes that are peaks. So this generates for you a rugged structure, right? And I promise that there would also in this case be a relation to the to spin glasses. So if you go through this, you can see that this becomes a kind of Hopfield model. So let's just say the phenotype function, the fitness function is minus z squared, right? So then this is, and then I put a minus sign so that I get a Hamiltonian, and then I transfer to plus minus spins instead of the 0, 1 spins. And then I get a kind of Hopfield model with a random or with fields, right? If I now assume these AIs are somehow random variables, then I get these Hopfield type couplings with random fields. And this model was actually studied by Nokura. It turns out to be an anti-ferromagnetic Hopfield model, right? So the way fitness maps to energy here means that this that the energy is minimized when the spins, in some sense, avoid the pattern. And it's also closely related to the so-called number partitioning problem, where the problem is essentially to take these coefficients and partition them into two sets which are as close as possible to each other in size. Okay, so this is the model. So why are we interested in it? And this, okay, I think I can at least sketch this. So it turns out, let me maybe give you the message first, right? So what we show is that fitness landscapes that are sort of constructed according to this principle have a very peculiar basin structure, and they are in some sense more accessible than typical landscapes. We call this the accessibility property. And unfortunately, the path to showing this is a little bit involved, but you know, if you're interested, we can discuss it in more detail later. A key starting point here is to use the isomorphism between the hypercube and the power set. This only works for the binary case, though. So this whole, you know, this discussion will now be for the binary case only. For the multi-allelic case, it can be generalized, but one needs to work a little bit more. But for the binary case, it's pretty obvious that instead of looking at sequences of zeros and ones, you can look at the sets, for example, of the sites which have a 1. Right? So then the 0 string is the empty set, and the 1 string is a full set, and so on. Right? So that's clear. Now the set representation allows us to think about epistasis in a different way. And this is the concept of, sorry?
What's a power set?
Just a set of subsets. So just a set of subsets of the sets of the elements 1 to L. Yeah. So this leads us to the concept of universal epistasis that Christina Krona and I came up with a couple of years ago. I should say, for lack of better name, I think the name hasn't really stuck, but since global and all kinds of other words were already taken, we said we call it universal. So what does this mean? So this is the following relation. So the idea is the following. You think about mutations as sets, right? So your mutations are just like a bag of stuff, right? And now you ask, so suppose I add a bag of mutations to two different genotypes, which are not in those genotypes before, where one genotype already has more mutations than the other, right? So you look at the fitness effect of adding a certain set of mutations in two different backgrounds, where one background is a subset of the other, right? So you take two backgrounds, one is a subset of the other, you add the same mutations to both of them, and then when there is a kind of strict inequality between the change in fitness, we call this universal epistasis. And the relevant discussion here is for universal negative epistasis. And we then, after
you've got your coefficients AI are all positive here.
Okay, so right, so
otherwise,
no, yeah.
How you choose?
I'll get to that. So if in the example that I showed, the coefficients were all positive. If you assume that, then this follows immediately. If you don't assume it, you need to rotate the hypercube, and then you have a different basis. You're, as always, ahead of me. So what we realized after a while was that this structure was actually known, and to do that, it's useful to sort of rewrite these sets a bit. So we can basically define the set A to be sigma and the set B to be sigma, the union of sigma prime and tau, and then you can write it in this more symmetric form, where on one side you have the fitness for the union and the intersection of two sets, and on the other side you have the sum of the fitnesses of the sets themselves. And this is known as submodularity in discrete mathematics, right? So this is a property of submodularity of set functions. And so now, with Daniel, we're writing a paper about this, so now we're trying to call this submodular epistasis. We'll see if that works better than universal epistasis. Anyway, this is an interesting property. It arises, for example, for this case of global epistasis. Yes?
Would you
compare and contrast this to diminishing returns, please?
It's very similar to diminishing returns, but it's sort of, you see, the point about this, of course, it is diminishing returns in the sense that the effect decreases, but of course, the effect doesn't have to be positive or negative, right? And it's also, so these landscapes, usually diminishing returns, you're thinking about some function that is only a function of the number of mutations, right? But these are, multi-peaked landscapes, and they can be very complicated. But nevertheless, when you do this comparison, you always have a certain inequality. But it is, of course, similar. And that's also why we think it might be biologically reasonable in certain contexts, right? And that's also why it actually appears in discrete mathematics, because many optimization problems are, by nature, have this submodular structure. Okay, so
sort of like convexity.
It's a kind of convexity, yes, indeed, right. It's a kind of generalized convexity or concavity, and so as Daniel already remarked, so if you look at this little example that I showed you, the one-dimensional model with positive coefficients, then this follows immediately, right? Because then you basically here you're comparing sigma and sigma union tau. This is the function. Now, if your function is concave, then you can just pull it out, and if you have the positivity of the a's, this follows, right? So this is one way of generating this, but as I said, you don't actually need it. You can relax the positivity, and it's still true, and it's it's sort of true for larger classes of landscapes as well. Now, why do we care, right? So this is now the last point. So it turns out that this submodularity has important consequences for accessible path, right? So let me recall, I said this already, genotype is accessible, or there are these accessible paths where fitness is increasing, right? So this is what we had already seen. And this is a property that Van Das discovered in a specific model in this class. We say that a fitness landscape has the subset superset accessibility property if any peak is accessible from all its sub and supersets along all direct paths, right? So you have a peak, it's a genotype, it has certain subsets. Supersets are just sets that contain the peak genotype. And it's accessible along all direct paths from any genotype in these sub and supersets. So the adaptive basins have a certain minimal size, right? If you sort of sum the subsets and the supersets, you find that there's a lower bound on the adaptive basins, right? So you cannot have adaptive basins that are very small. Also, these landscapes contain at least two genotypes from which all peaks can be reached, because these are the genotypes that have no mutations and the genotype that has all mutations, because it's a subset and the superset of every other genotype, right? So this is the property of interest here, and this is just an illustration, right? So this is one, again, a hypercube, and here we have two peaks, so the red and the blue one. And these arrows now show those genotypes that have to be in the adaptive basin because of the accessibility property, right? So these are all the subsets of the blue one, and these are the supersets of the red one, for example, right? So basically what this means is that the peaks have sort of a region around them which are fairly flat and where the evolution is sort of forced to go towards these peaks. And now what is the connection between the two? Well, we can show that submodularity implies the accessibility property. This is, again, a very simple proof. So if you know that your genotype is a peak, it means that if you add a mutation to it, then the fitness goes down. And if you subtract a mutation from it, the fitness also goes down, right? So this because you're a peak. Now you look at a subset genotype and a mutation that you want to add to it, right? And then you look at the corresponding fitness difference, right? And then because of the universal negative epistasis, this fitness difference is bounded below by the fitness difference at the peak, but that you already know is positive, right? So therefore, adding this mutation to sigma prime will also increase fitness, right? So this is a very elementary statement. We published it in this paper with Daniel in 2024. And then, when we sort of learned about this connection to submodularity, we found, of course, that it was also not new. And the earliest reference that we traced it back to is by a Soviet scientist now with the name of Cherenin, who apparently, I couldn't read the paper because the paper is in Russian, but apparently it was a study, and this is particularly touching for someone coming from Germany. This is a study on the optimization of the train schedule in the Soviet Union. Right. But it is a property, so it was rediscovered subsequently in the submodularity concept. It is a property that is known. I don't know that it has been used very much, but in the context of these fitness landscapes, this sort of gives an interesting relation between the structure of global epistasis and the accessibility structure. Okay, I think I'm actually done. So, and I think I'm pretty much on time. Okay, let me just this last thing. So, as I said, this gives you a lower bound on the size of the adaptive basins. But in fact, and this is this lower bound, right? So this is for a system of size 8. This is showing the distribution of the adaptive basin sizes, and these are the lower bounds. But of course, you see that the typical basins are quite a bit larger. So we don't know at the moment whether this property really increases the basins in a significant way, right? Because as I showed you in the previous part of the talk, even a random landscape has very large basins. So this effect might not be that large at all. Okay, so let me then conclude. So I've shown you a couple of examples of experimental fitness landscapes that have motivated the study of these random landscapes. And so the goal is to characterize the landscape topography. We could also say complexity. Yeah, I mentioned complexity here. So for me, these are all discrete spaces, but for me, complexity is just, the exponential growth rate of the number of peaks. I mean, that we can still define, accessibility and epistasis. And I think this is, interesting field to work on. I want to acknowledge the main collaborators. As I said, this started with Ariane de Visser. I have a long-term collaborator in Seoul, Suchan Park, and Christina, who is here. A number of postdocs have been involved in this, most recently Saverio Rossi, and also, of course, quite a few students. Okay, so thank you for your attention.
Questions, comments?
So perhaps a general question, perhaps too general, is how strongly statistics or observables, such as, for example, the size of the adaptive basin, depend on the tails of the distribution? In the sense that if you're looking at the random landscape and, for example, the other extreme, which is a global epistasis landscape, might get things.
Yeah, okay. Let me first say, of course, in the random case, nothing depends on the distribution, right? Or I mean, the distribution of the fitnesses doesn't matter, right? Because everything we talk about are order relations, right? So changing the fitness distribution has no effect, right? So then
So the question is, if you look at another landscape, such as a global epistasis landscape, the question is basically how well can you approximate its statistics with results, for example, like the one that you showed for the random landscape?
Yeah, that's I think pretty much the same question as before. The answer is I don't know. I mean, I think this is, we have the solid result for the random case. We have partial results for these structured landscapes, right? So for example, so for the global epistatic landscape, we have this lower bound, but you can see here that this lower bound is not possibly not particularly informative. So, and we have some results. I should, I can sort of mention that. So for the other structured landscapes, we have here. So what we can actually prove for the NK model, for example, one can prove. So there things depend a bit on how you scale this interaction range with the length of the sequence, right? If you scale the sequence length to infinity at fixed interaction range, then we can actually show that in the NK model, there are no accessible paths of length of order L at all. So it has to do with the sparseness of the interaction graph, and this will sort of partition the landscape into regions that you cannot communicate between with accessible paths, right? So here, in that case, at least, introducing structure actually makes it much less accessible, which is not what you would naively expect. In this case, it's not so clear. I mean, here, the main effect, as you might imagine, you're basically introducing a kind of fitness gradient. That, of course, makes it easier to go uphill in the direction of that gradient, but it will strongly reduce the sort of indirect path, right? Because these indirect paths that go up and down, there will be much fewer of them. So on balance, how that affects the accessibility, we don't know.
Yeah, just a sort of comment that makes connection with this morning's talks. I mean, from the evolutionary purpose, once one gets away from a very small number of sites in a single gene being important, then one is in a limit where it's nowhere near exploring all of the space, the evolution. And so then one sort of wants models where the total length is large, but the total number of mutations that is a small fraction of the total length. And then there's classes of models which Tish Agarwala particularly had looked at, where you do analogous to what Gerard was talking about of the where you take a random Gaussian one with some correlation structure, and it depends on the distance, the Hamming distance between two genomes. And then there's a lot of sort of whole families of them which actually include some of these. And rather interesting behavior. We only know little bits about them, but they're, I think, in some ways, they're more biologically motivated in the sense of they're intrinsically thinking in terms of large genomes, or even just large number of amino acids that matter.
I mean, maybe one thing that I also thought about this morning in the talk of Gerard. So you emphasized that in your landscape, the problem might not be so much that you get stuck at local peaks, but that it takes so long to find any peak at all, right? So this findability. So there is a kind of analogous concept here, which is related to this this distinction between what is it? Reciprocal and simple sign epistasis, right? So if you have, so I said in order to have multiple peaks, you need to have reciprocal sign epistasis, right? Now suppose you have a fitness landscape that only has simple sign epistasis, but lots of it, right? Then you can actually, so then there's only one peak, right? So the peak the ruggedness is not an issue, but then you can show that, if you organize things correctly, you can have situations where finding this peak will take an exponentially long time, right? So you get these paths that sort of keep wandering around and that never find the peak, right? So there is a kind of issue in terms of findability also in this setting. So that's yet another class of landscapes that I think haven't been looked at systematically.
Yeah, just a comment about what's now called super modularity, right? One can extend that to the case with several alleles very naturally, but then there's this discrepancy between Euclidean distance and Hamming distance, since if we call an allele 1 or 2, has nothing to do with distance, right? It's just a name.
We should maybe discuss this. So we struggled a bit how to generalize this to multiple alleles.
Well, but just a very short comment. A nice relation we had to staircase triangulation doesn't work if you have several alleles.
I see. Yeah, I should have said that. So this universal epistasis was first discovered in the context of this classification of fitness landscapes in terms of triangulations of the full cube, right? Not the hypercube, but the full cube where you put in planes and so on. But this is sort of another topic that Christina is an expert on. Yeah.
So in addition to properties of the landscape, I guess there's also the process by which the population moves.
Yes, yes, sure.
So can you say things about, for different classes of landscapes, which types of selection will be efficient for sort of finding maximum faster? For example, I can take the biggest step possible, or I can take the smallest step possible, or I can weight them by some.
Yeah, I mean, this is so. First of all, it's a good comment, and it's important to emphasize that these are rather different problems, right? So here I only talked about the structure. Now, once you introduce dynamics, for example, these huge adaptive basins, right? Of course, this doesn't mean that typically, and it wouldn't even make sense. But suppose you could interpret this finding in the following way. You could say, okay, I know the global peak of my landscape is reachable from 50% of all genotypes, right? That, of course, doesn't mean that with 50% chance, if I start at a genotype and I do an adaptive walk, that I will actually get to the global peak, right? So these dynamical paths have a completely different statistics. I mean, the accessible path that I defined, they are, of course, all in principle possible trajectories of an adaptive walk, right? So if the adaptive walk made the right decision in each step, it could cover this, but these paths have very low weight, right? So the typical adaptive paths are much shorter, and they will end up at peaks that are typically close by. And then about those, of course, you could then ask, what is the effect of different types of dynamics? And there, again, it depends a bit on the landscape. I mean, I think some landscapes, the fitness that you reach under greedy dynamics are higher than those that you reach under random dynamics, and for others, it's the other way around. So this is more complex.
Just adding that, I think this is very relevant for the evolution, because it depends, does it pay to have one big population with everyone competing, which will be very good at the greedy parts, or having a lot of little competition,
sure,
but not many competing,
yeah,
which will do better exploring.
Yeah.
And if it's sort of close to not much epistasis, then it does better to bring everything together. But I think generically, it's much better to separate it and not be greedy. Certainly, the whole class of landscapes where one can be not being greedy is much better.
All right, so I think we should take your last 12.